Cornell and obtained his bachelor's degree in 1999, and he finished his schooling and achieved his doctorate from Harvard in 2004, and he actually became a faculty member of Cornell in 2008, and some other milestones in his life that he's kind of achieved is he won the award, the Guggenheim Fellowship of Natural Science for use in Canada. So that's just to present it. So the content of my lecture, it mainly just revolves around the central dogma of a cell. The central dogma is basically just the organelles and locations of a cell that are needed for DNA to be synthesized into proteins, and then those proteins to be localized into where they will be most helpful to the cell. And that process is pretty linear. It just goes from DNA being transcribed into RNA, and RNA being translated into proteins, and those proteins making their way to the Golgi complex, which is kind of like, as a simile, I guess, like the grand central station of a cell, where proteins are taken there and they enter vesicles, and these vesicles are carrying things like macromolecules such as proteins and lipids, and these vesicles are then being transported to places elsewhere, either in the cell or out of the cell. And so these two overarching topics are what Chris Fromm based his entire lecture around. And so continuing on, there were even more specific topics that he got into, but before going into those, he asked two main questions, those being, how are decisions made and how are different pathways regulated? And so when considering these questions, he was only looking at the Golgi complex and applying these questions to the Golgi complex. And so he first started by explaining that there are two families of these things called guanosine triphosphates, or GTPases as he referred to them, that regulate the trafficking of vesicles in the Golgi complex. And the two main guanosine triphosphates that he talked about were ARFs and RABs. And ARFs, they control incoming vesicles from the top of this diagram of the Golgi complex. All those circular bubble-looking things at the top of the Golgi complex are vesicles, and they are carrying macromolecules such as proteins and lipids. And so he explained that ARFs are basically just signals that control when a certain vesicle, I guess, attaches itself to the Golgi complex and releases its proteins or lipids or whatever it's carrying into the Golgi complex to be processed. And the RABs are, they control outgoing vesicles at the bottom of the Golgi complex, where these proteins or lipids are being transported out again into vesicles and being located to where they will be most helpful to the cell. And even more specific topics that Dr. Fromm went into. So clearly he was not satisfied with explaining how the Golgi complex is regulated. So he delved deeper into how the regulators of the Golgi complex are regulated. And so he went on to explain GEFs, which stands for a guanine nucleotide exchange factor, and they are proteins that activate GTPases by stimulating the release of guanosine diphosphate to be allowed the binding of GTP. And so some GEFs can activate multiple GTPases while others are specific to one, and that is just a structure of a GEF. And so really this whole lecture is basically just a linear path of regulations and signals that are sent to achieve a common goal throughout one large organelle in a cell. And he really attacked every detail when explaining that. And so another specific example that he gave is TRAC2, which is a specific name of a GEF. And this specific GEF activates a specific RAB named RAB11. Now RAB11 is special because it regulates vesicles carrying very important macromolecules such as membrane-binding proteins for either intercellular communication or facilitated diffusion, and motor proteins for transport. And this is a diagram of these facilitated diffusion channels that might be transported to the membrane of a cell. And this is very useful GIF of a motor protein, one of my personal favorites, if I must say. And so why this is important? This is very important because the field of biology is always changing and it's always advancing because the study of biology is ongoing. One of my favorite examples of this is the cytoskeleton. So the cytoskeleton was discovered fairly recently, where it's just been recently added into textbooks, and so this is just one example of how biology has been changing in the past few years. And another reason why this discovery is very important, the Golgi apparatus and its regulations, is just for the general understanding of the building blocks of life that are cells. And so the work that Dr. Chris Fromm is doing at Cornell is very important just for the sake of science in general. And so my thoughts throughout the lecture were, this is way beyond my understanding of biology, and so going into that, I just tried to keep an open mind as to what I could get out of it, and apply my past knowledge of biology as much as I can, but also understanding that I do not understand. And that is all I have, and are there any questions?